Welcome to Own Your Heart Health. I'm Dr. Regina Druz, your holistic cardiologist. This week, we will dive into common heart health concerns, uncovering root causes, and unpacking scientific discoveries and controversies. The information provided does not constitute medical advice. Please contact your healthcare practitioner before making any changes that may impact your health. Hi, everyone, and thank you for listening to Own Your Heart Health with me, Dr. Regina Druz. So I have absolutely a fascinating guest today, and I feel like today we're going to walk on the wild side. I kid you not. I'm with Dr. David Scieszka, who actually is the CEO of something called Vertical Longevity Pharma, bringing us some news on creating a vaccine. Just listen to this before you turn me off because you're against vaccines. I want you to hear this in its entirety. Creating a vaccine against the number one disease that kills men and women pretty much in every country, and that's atherosclerosis. So I have never heard about a vaccine for atherosclerosis. I have no idea what it is. I have no idea why we would, certainly we would love to prevent atherosclerosis, but how do you do it with the vaccine? I have no idea. So Dr. Scieszka, David, how did you grow up to be who you are today? Just fill us in, connect the dots for us, please.
Yeah, thanks for having me. So let's see. I started off my scientific career in the Army, which most people would not expect, as a psychological operations specialist, or Psyop. And what we were doing is basically, in times of peace, we're census takers. So we would go into friendly territories and we would say, how are you doing? What do you need? Do you need a hospital? Do you need a library? How is your food supply? Do you need toothbrushes? That kind of thing. And we would also take a name, age. And I found that people in rural areas were aging at a much more accelerated rate than people in cities. And access to healthcare was really the main driver of that I could figure out. And so that to me was this real world example where aging is not set. Aging trajectories can be altered. And so I thought, well, I'm going to go figure this out, as a naive young man does, or a young woman does. And I got immediately hit with how complicated this process is. So I did the biotech degree undergrad, started doing computational biology, because I figured that this was going to be a complex systems-based problem. And it turns out that it is. And then I went on to do a PhD in biomedical sciences focused on aging, and an MBA when we started really realizing that this vaccine, during my postdoc, I generated all of these data, and I realized that this was going to be a thing. We needed to understand how to run a business, and that was not part of the undergrad training or any of the PhD training. So that was a tack on at the very, very end there. But yeah, that's a long story made long. Sorry about that.
No, it's a fascinating story. And, we'll come back to the vaccine part because I'm still stuck in the Psyops, so if you could explain to those of us who are not familiar with military sort of setup and operation, what are Psyops? And I understand what they do in peaceful times, but what do they do in times which are not so peaceful? So what is it?
Yeah, let's see. A very famous example would be, historical example anyway, would be blowing up fake tanks, so balloon tanks, really, on the shoreline of England, so that Germany thought that England had this massive tank battalion. That was a psychological operation. It was to move the attention somewhere that we wanted it to be, instead of maybe on a landing where there actually troops were. So if they blew up a tank balloon, nobody gets hurt. But if they were focused on the beaches where people were landing, then people are in harm's way. There's a little bit more nuance to it sometimes. So we do the flyers. We drop flyers from C-130s informing people of anything. There's also trying to understand where terrorists are. So you can go in and converse with local populations saying, we're providing you a hospital. Could you provide us some intel? We would really like to take these bad guys away from you, out of your life. If you can help us out, we can help you out too. So that's many different functionalities there. I like to kind of think of it like upgraded infantry. We can shoot and move, but we can also think very on deep and difficult questions and try to form solutions to them.
Wow. So this is fascinating. And I think it explains a lot to me how you transferred this sort of mindset into the field of longevity, right? So, from observation, sounds like observations on patients who lived in rural area. Were those specific rural areas, sort of, the parts maybe in the United States or elsewhere that don't have access to a lot of resources, a little poorer, they're like, we know that there is a lot of healthcare disparities for sure, right? We're a big country, and one of the things we hadn't really solved is how to give people access even to basic care, and that's a tremendous negative, I think, and we're sort of behind a lot of other countries that figured this part out. But so how did that sort of translate into wanting to pursue longevity? Like, you could have gone another route, right? You could have done a political stuff, and advocated for more resources and become an economist. So how did that sort of transition into the longevity vertical?
I actually thought I was going to be a politician. That's funny you mentioned that. I wanted to become a politician because of the access to healthcare I thought was an immediately solvable issue. But that, it turns out, is something that I think we're going to be addressing for many, many decades to come. I mean, we still have food deserts in the US where there's
Right.
nothing but fast food and no health, no organic food, that kind of thing, for miles and miles and miles. And so that's contributing to the overall health disparities, diabetes, well, cardiovascular issues too. And I used to work in the College of Pharmacy in environmental toxicology, so we would take a look at these population-wide studies sometimes, and it's striking how backwards, or maybe behind the times, as you said, that we still are in the current day. Yeah. I couldn't actually
Yeah.
do the political round though, so I ended up doing the science instead.
You're doing the science. You took the easier of two paths, which is the biotechnology PhD. But it's fascinating because I just read a study the other day. It came out in Journal of American College of Cardiology on the impact of ultra processed foods on cardiovascular events. And there is dose direct dose relationship, right? So basically, each extra serving of ultra processed food increased risk by about 5%. But what was fascinating about it, two things actually, is that number one, the whole classification of what constitutes ultra processed food it's not very straightforward because it's called NOLA classification, and it doesn't really look at the nutrients, it looks at the degree of processing. So like you could have fried fish and soy milk, and that would be ultra processed food, right? By NOLA criteria. And then there was an interaction which was very troubling, but I think we're seeing it clinically, and you probably saw it in your earlier days, Psyops, interviewing patients, is that there is actually an interaction between ultra processed foods and race. So Black patients didn't have 5%, they had 6% or more. So there were factors that were interacting even within that, like, not a perfect system. So, atherosclerosis. Tell me how. So you went into biotech. You said, okay, I'm gonna solve this. I'm gonna help people reverse aging or age a little slower. So what did you find in the lab? Like what were these discoveries that kind of propelled you later on in this vertical longevity?
Yeah, that was actually a long amount of testing that we did. So not specifically in the vaccine itself, but I've been in this field for 14 years now. So we've been testing. And to your point about ultra processed foods, we actually know that a lack of fiber, increased sugar, and the fiber can offset some of the fat. But if you have a high fat, high sugar processed food, that does terrible damage. But you can try to offset it with some of the fibers. So that's like real world context that doctors tend to take a look at. And so
Mm-hmm.
we took a look at these longevity therapies in real world context, because it turns out that mice living in a sterile environment, of course, mice aren't humans, but it gets even worse if they're in a sterile environment because we don't live in that world. We are affronted
Right.
constantly by bacteria. Somebody smoking on the street, you roll your windows down on the freeway, you get hit with a bunch of diesel exhaust. I mean, these are real world contexts that make a difference. It turns out when we were testing some of the longevity therapeutics, we saw heart failure after
Really.
wildfire smoke events. So these things called senolytics, but I don't need to necessarily get into exactly what that is yet, but it's a longevity thing. Those can cause heart failure after a wildfire event. And they are being sold over the counter right now. So
Really?
this epidemiological study, it's hard because of the statistics to actually get that signal high enough to say that these are damaging people instead of helping people. And it's context dependent as well. So they'll continue to be sold. It's just a lot more dangerous, it turns out, than people expected. And so that was kind of the impetus for our future, our current vaccine, and now the future direction and future work at Vertical Longevity.
Interesting. So what is the concept of vaccine against atherosclerosis? What is it actually based on? In other words, what are we vaccinating against? Because, being a clinical cardiologist, I think of vaccines as like, okay, chicken pox, right? It's an infectious agent. It's obvious. We know what it is. But atherosclerosis is a very complex disease. Yes, we would like to execute what's called primordial prevention. In other words, get to the point where people don't even have risk factors for atherosclerosis. But honestly, atherosclerosis, in my mind, and I said it numerous times, I'm going to say it again, that is aging in my eyes, right? Because aging is vascular. That's a system that shows us all the damage, all the influences, and, also participates even in other diseases like cancer, for example. So, with aging being vascular, how do we even conceptualize why vaccine? What are you doing exactly? I guess that's my question.
And so in the cells that line the inside of the arteries and veins, they're called endothelium. And those cells, they can transform into this senescent state. And that's a state of cell cycle arrest. It's pro-inflammatory is actually the key point, though. And it's no longer functioning as it's supposed to. So vascular tone is decreased. There's also signaling that's no longer functioning appropriately. And then as you can imagine, the inside of your artery needs to be a very, very tight barrier, keeping things in circulation that are supposed to be there and stopping circulation from entering outside of the vein itself. But when senescent cells permeabilize, or when they transform, they permeabilize that barrier. They open up gaps between them because they can't hold on to their neighbors as strong anymore. And so this is actually, there's a growing body of evidence to support this, but it's not as well known in the cardiovascular field. Branch points we have known for decades are major hotbeds for atherosclerosis. But this is a mechanistic explanation of why. Because they permeabilize and turbulent flow slams the LDL in between these gaps. So then it can initiate these plaques, and then the senescent cells themselves are pro-inflammatory, preventing the macrophage-based the immune cell-based clearance of the plaques themselves. So we're starting to get into this realm where aging intersects with cardiovascular biology, arterial biology, pulmonary, kidney, liver. Everything that touches blood is receiving inflammation from these senescent cells. And so then by clearing out, that's what the vaccine targets, is these senescent endothelial cells. And by clearing them out, the entire body received a therapeutic benefit in preclinical studies. The entire mouse body received a therapeutic benefit, including the ability to prevent plaque. And we're testing now in primates in our upcoming study the ability to fully clear plaque, because we believe that that's going to be mechanistically plausible as well. So we're actively going to test it.
Wow. So this is fascinating and this is complex. So let's unpack it sort of step by step, right? So we, people may or may not, our listeners may or may not know that our bodies have living cells and dead cells, but in between living and dead cells is this intermediate stage. And sometimes, in my upcoming book, Longevity Decoded, they will read about it. It's called zombie cells, right? So these zombie cells are sort of, horror movies. Who are the zombies? They sort of, they are dead, but they behave like they're alive and they're very scary and they are, could do a lot of damage. So that's exactly what those senescent cells are, right? They're zombie cells. They are actually, you would think that they're just sitting there and maybe occupying some space, but that's not how it works, because they actually send signals, right? Sort of like, oh, zombie, right? So they send these signals to the surrounding tissues, and they promote inflammation. And our inability to clear those senescent cells, that's one of the hallmarks of aging, right? That's how aging actually happens. We can no longer, or we are not as efficient, and eventually we cannot clear these senescent cells, and the damage accumulates and sort of becomes compounded. So what you're saying, right? Am I correct so far? I'm just trying to give like kind of a bigger picture here.
That's a fantastic explanation. And they do transform their neighboring cells into senescence as well, which is hence the zombie. Yes, absolutely.
Since the
correct.
zombie effect, right? So they actually, they are major influencers. Initially when I first was learning about it, I thought, it's probably not such a big deal, but they're major influences and very dangerous. And so what is happening under those circumstances, if I understand you correctly, is that they actually, these senescent cells, and first of all, let's step back a second. Endothelium is the inner lining of our blood vessels. It is only one cell thick. Every single blood vessel in the body, even capillaries, has that lining, right? This is us. This is who mammals are, right? And endothelium is actually the largest organ in the human body, right? If we were to take all of those little cells and spread them around, we would have, a football field or even more, depending on where you read. So endothelium, if I get you correctly, could become leaky, right? Sort of like we have leaky gut, sometimes we have leaky brain, but here we literally have leaky vessels. Does that sound right?
Yes, exactly.
Excellent. And so how does vaccine actually work? What's in it, and it sounds like you injected it in mice, and, the next step are primates, but what's in it that, what mechanistically is actually happening to help people eventually, for now it's just animals, to help people to clear out those senescent cells from their blood vessels?
Yeah, and that's part of the scientific process. So we did not. Senescence is incredibly complicated biology. It's one of the reasons I love it so much, but it's not always the same expression profile. So when the cell transforms senescent, they change the amount of genes that they're activating and proteins that they're creating, but not always in a consistent fashion. But thankfully, in the endothelium, once they transform senescent, just like a flag, they pop out the surface marker that then we can target with a vaccine-based antibody approach. So we have the immune system generate antibodies against that surface marker, and then your immune system clears out those cells for you, just like it did when we were younger. The ability, like you said, to clear these senescent cells used to be relatively robust, and that declines with age, unfortunately. So we're re-engaging the immune system to be able to work like it did back when we were a lot younger.
Interesting. So at what age, do we have evidence that our ability to clear those senescent cells with our own immune system without any additional stimulation, at what age does it start to decline?
I would say as soon as growth has really stopped. So there's going to be different ages for different people, but there's around 25-ish, if there would be a rough number. And then the process is actually accelerating. So it'll start off very, very slow, but it's exponential. And so in the case of senescent cell accumulation, because senescence begets more senescence, you're going to have one cell transforms into two, transforms into four, transforms into eight. And so that's why it's an accelerating process, unfortunately. And the body's ability to clear that out declining does not assist the situation whatsoever.
Yeah, so it's interesting because, speaking to so many researchers in the field of longevity, I have to say it's a little bit disheartening to learn that this process starts in late 20s, early 30s. I was sort of hoping like, okay, 40, 50, yeah, that's what we'll expected, but that's not the case, actually. It starts much earlier.
The process itself starts earlier, but this is actually a fascinating topic of the what is aging. So there's statistical evidence population-wide to show that there are two acceleration points of aging around 40 and around 60. And it's going to be different for everybody. But that is, in my opinion, as a systems biologist and computationally, there is an equilibrium of the body where everything's talking to everything else, interconnected, But then a lot of these support pillars start to crumble. And so then this equilibrium right around 40 has to shift. And now it's an acceleration of aging because something broke. Something was no longer able to compensate to support something else. And that broke. Now there's a new equilibrium, an acceleration of aging, and damage is continually occurring until around 60. And then another breaking point happens. And then we have this additional acceleration. So we can see this physiologically, but we can't necessarily point to something saying that's the cause of breaking. It's senescence, it's environment, it's genes, it's it's everything around us that's causing this.
It's fascinating because I was reading about a woman named Maria, and she lived to 116 years. I think she was studied by Spanish investigators. She's abbreviated as M116. That's sort of like for 116 years. And what they figured out is that on some of our sort of internalized conceptualization of what is aging, which is what you're describing, right? Based on these hallmarks of aging that are more of a conceptual framework, but we do find more and more evidence that it's a real framework. She was kind of the opposite in a lot of things. For example, her telomeres were extra short. And so they have conceptualized that actually protected her from cancer because her cells were not necessarily dividing that fast. So it's just fascinating that what we think we know, people come out there and we see sort of like, wow, it's not that clear cut. So senescent cells, I'm fascinated by these. Do they occur in every organ? And would you say the second question is that vascular senescence, because, let's face it, our blood vessels do a ton of work from the moment we become a human being to the moment we are passing, exiting our physical domain. Blood vessels are hard at work. They never stop. The heart never stops. So is vascular senescence the earliest in this aging phenomena? And obviously other organs will follow. Is there some sort of a pattern there? You alluded to these sort of aging thresholds that science is looking at right now. So how does that all connect?
Yeah, we do know that every cell type can transform senescent. Unfortunately, that was actually debated for a long time, and there was wonderful work done by an investigator out of UC Irvine, and she said neurons can transform senescent. And a bunch of old guys in chairs were saying, no, we haven't seen any evidence for that. And she just kept at it, and she was right. She was vindicated. It is absolutely true. Neurons can transform senescent. That was one of the last cell types that we were unsure about. Cardiac cells can transform senescent, liver cells, everything can transform senescent, but to a different degree, like you mentioned. I would love to see those studies if we had any human-based data available, but in some cases, you would require some decedent tissue, and not every age range has that available outside of a disease type scenario, because we're hoping to capture the normal occurrence, the normal acceleration. I don't know if those data are out there. I would love to see that study though. But I bet you're right. I bet that there's going to be, based off of just the amount of stress that endothelium undergoes on a constant basis. I bet liver would probably transform a lot of senescence, but there's also internal cellular mechanisms that can recognize when a cell, like in the liver, is defunct and dysfunctional. And so it tries to kind of monitor itself, be its own self.
To clear it out.
Exactly. Yeah. More so than perhaps the endothelium, more so definitely than than the heart itself, because there are no stem cells to replace the heart. So it would be detrimental to clear those.
Yeah, exactly. So how does, you described sort of high level overview of how the vaccine works, right? The senescent cells have a marker, a molecular marker. Is this marker the same that normal cells have as well? Or is this marker specific to these senescent cells? So when, let's say, vaccine is injected, the immune system will make an antibody only against that marker and will not damage a healthy tissue.
Exactly. So it exists as a functional peptide only in the context of extreme bone compound fractures. So it's associated with a little bit of bone remodeling, but we've talked to osteo experts on this too. It was a major concern for us, and I could tell it will be a major concern. So we brought them the data showing where this protein exists, what its normal function is. And they said, we actually don't expect there to be any problems at all, because the amount of endothelial senescence that's just around. And then this is also on some foam cells. So we're going to be clearing out foam cells inside of the plaques too, which could be a very good thing. They said with that amount in the regular just existence of the body, we do not expect there to be any problems. We'll do the study for you down in Australia. It's going to be a fantastic study down there, but they don't expect to see an issue with it. We will still test it because I'm obsessed with safety. Even if the FDA does not require it, I'm going to test that. I'm not going to put this.
So what's the name? What's the name of that protein? So it's expressed in bone tissue, right? In bone tissue cells, if I understand you correctly. What's the name of that protein?
I could talk to you after. I could tell you afterwards.
All right. So that's basically, but there is, you want to make sure from the safety perspective that the healthy tissues are not impaired. So what have you seen in mice so far? Give us some hope because, while we know that mice are not people and people are not mice, the truth is a lot of longevity and, geroscience, as it's called, experiments have been executed in mice. So, it's one of those models that, we'll always go after. So what have you seen in mice experiments so far?
Yeah, we actually saw a therapeutic benefit in every single organ and system we were able to monitor. So that was one of the reasons I left academia, was because we studied it. We stress tested this vaccine using the same metrics that we saw cause heart failure in mice, was the senolytics, statins, and quercetin. We ran echocardiograms on those mice, and we saw heart failure before it actually turned into heart failure. So we ran echoes after vaccination, and we actually saw cardiac rejuvenation in the opposite direction that statins and quercetin was showing. We studied the pulmonary space. We saw rejuvenated pulmonary function in the form of breaths per minute, tidal volume, multiple different functional outcomes. We saw prolapse prevention, which is an age-related organ. There's also hair loss prevention. We expect to see chronic kidney disease reversal because senescent cells are a major driver of CKD. There's probably going to be vascular dementia effects. We haven't shown that, but mechanistically it's plausible. And again, this vaccine is going to assist every organ that touches blood. That's going to be one of the major outcomes, is that it's a whole body rejuvenation. And to the human case, there actually has been a previous 3 or 4 clinical trials using antibody drug conjugates, which is a chemotherapy connected to an antibody, against our exact protein. And those always passed phase 1 safety in humans. They were going after breast cancer, though. So they passed phase 1 safety. They were unable to cure breast cancer, so the clinical trial stopped. We know what level of antibodies we should be able to generate to reduce that to reduce that risk window down to, from a statistical standpoint, a near zero likelihood of a serious adverse event. And that's informing our dosing strategy. So we know it exists in humans. We know it's a cancer-associated protein in humans. So we might have anti-cancer effects too. I mean, I know it sounds like one of those magic pills, but I mean,
it sounds very, very sci-fi, very futuristic.
I appreciate it. Yeah. And hopefully that's not off-putting, because it does sound fantastical until you start to see the data. And once those data come out, and instead of seeing negative outcomes, everything was positive, that was the impetus. We couldn't let people's lives expire because we did not act. We had to take this and try to commercialize it. We had to try to save lives. And then that's how I met my CSO, because we are mission aligned on that. Yeah.
Sure. So it's fascinating because I'm just thinking about those little mice and little tiny echo probes. And, I do echo on every single patient when I see them in person with my little handheld transducer. But, the reversal of cardiomyopathy, which is what you're describing, the weakness of the heart muscle. And obviously it has a clinical association with heart failure as a syndrome, but just the fact that it's capable to reverse itself to a degree where heart failure reverses is remarkable, because, at least in human heart failure, we know that there is sort of this discordance between the ejection fraction and the clinical syndrome of heart failure, because humans are a bit more complicated. But obviously, normal function, if it's been diminished, is a great progress, and, a little bit harder for heart failure with preserved ejection fraction. So, David, how in the mice studies, was it sort of one and done? You injected this vaccine once and you had seen the impact, or is it something that will require repeated injections? Like, in other words, how much of that immune system stimulation must you carry out for the effect to be sustainable, at least over a reasonable period of time, let's say a year,
Yeah, that's a really good point. And thank you for the question. So it was a 3-dose series, and that's to ramp up the immune system. But we're using a virus-like particle, and I just wanted to hit on a point that you made at the very beginning. Anybody who's worried about a vaccine out there, a lot of the arguments tend to be around the adjuvant. And regardless of how crazy I might think those arguments are, this has no adjuvant in it at all. It's recognized like a virus by your body. So we're circumventing any of the anti-vaxxer sentiment, potentially. Maybe some people will just be put off by calling it a vaccine, regardless of whether.
But it is a vaccine. It stimulates the immune system. So
Exactly.
Mechanistically, it is a vaccine.
Exactly. No alum, no adjuvant. It's just recognized like a virus. But yeah, so it's a 3-dose series, and we're trying to get strategically, we selected the virus-like particle because it can be produced so cheaply. We are on par with statins, basically, and statins are an incredibly cheap to produce and purchase. We don't want this to be one of those things that only rich people can get. This is not going to be stem cell therapy. This is going to be like, go to my doctor once a year, get a flu shot, and also hit me with that senolytic vaccine. And it's, free because we're going to get the payer CPT code for the payers to be able to pay this from an insurance standpoint. We want to make this broadly accessible for as many people as possible. And the 1-year time frame that will likely require a booster increases the safety as well. If you ever have an issue associated with this vaccine, which we don't expect there to be, but if that ever occurs, because humans are variable, just stop. And then without boosting, all of the antibodies will slowly go away. So this increases the safety as well.
Yeah. Interesting. So how much have you seen in terms of plaque regression and plaque reversal? I mean, obviously these are animal models, and again, mice and humans are not necessarily comparable, but it's a reasonable model to try it in. So what have you seen with the vaccine in that scenario?
Yeah, with the vaccine itself, so we're actually utilizing external biological validation. There are now 3 labs testing the vaccine itself in different vaccine modalities. We tested largely safety. A different group showed that senescent endothelial cell clearance targeting the same target can prevent plaque by up to 50%. And then another group showing senescent endothelial cell clearance can clear existing plaque by up to 40%. So that's a whole plaque that once was there, fully gone. That's a number of plaque removed. That's the 40% there. So it's true.
So
reversal.
Healthy endothelium.
Exactly.
The endothelium is regenerated. Like all the foam cells, all the calcium deposits, everything is gone.
And this is over a relatively short duration. So in the human case, it would probably take about a year. For this. And thank God, because it's a slow mechanism. You're not worried about the rupture or anything like that. If it was too fast, could be potentially dangerous. But yeah, 40% plaque reduction in such a short time. Imagine what's going to happen if people are on this for 2 years. We could have complete rejuvenation of the endothelium to a state where there's no atherosclerotic plaques anymore at all. Potentially, we have to show that, of course. But I mean, could you imagine?
What my mind right now is going 1,000 miles a minute because, in last year in August, I went to the European Society of Cardiology, and a company presented their research on CRISPR. And of course, CRISPR being gene editing tool, they went after familial hypercholesterolemia, genetic carriers of lipoprotein little a mutation, very damaging lipoproteins. And we all know as cardiologists that lipids are super important, but unfortunately, in the best lipid scenario, you're only addressing 30% of person's risk. 70% of risk, what I always tell my patients, this is what cardiology calls residual risk, a total misnomer because 70% is the majority, cannot possibly be a residual. I think they need to just flip it around because it's like we'll go back to 6th grade math and reacquaint themselves with residual versus, main thing. But the point is that we all know that residual risk is what actually shaping cardiovascular outcomes, right? And so even the connection, an interesting sort of mental construct there is that we know some of the more powerful drugs, like, for example, PCSK9 inhibitors, in some trials show plaque regression, but we're not quite sure if plaque regression actually leads to event reduction. Why? Because again, that residual risk, that endothelial dysfunction. So I guess what I'm interested in, let's say we are in the situation where it's a person who has, let's say, bad genetics, right? They have family history of coronary artery disease, early coronary artery disease, or lipoprotein(a), or familial hypercholesterolemia, and most of it is it's a rare disease in its worst form, but there are multiple genetic variants that are giving people an enhanced risk on the lipid side without even being FH patients, right? So you're a computational biologist, so you know this, the old polygenic risk factors. So would you think that patients like these, let's say vaccine goes on and gets approved and is safe, they would get a prevention shot? Let's say they're 18, right? Maybe they just have fatty streaks, which is what we know in the coronaries. Like, would they get a prevention shot not to form plaques?
That's exactly the beachhead that we're approaching first. We are mirroring the Pfizer playbook, actually, to go in FH first to try to prevent those major cardiac events. We're trying to prevent those because that's a meaningful clinical outcome. And that would feel incredible to save lives from our very first set of clinical trials. I mean, it doesn't get better than that. And so that's going to be our beachhead that we go after first, because I believe, I like to visualize things, and I believe in the FH story. There's a lot of endothelial dysfunction, and then by repairing those barriers again, you can have circulating cholesterol up the wazoo, and it's just not going to permeabilize. It's not going to get through those gaps. And so that's hopefully what's going on in the FH case. With the LPA, I visualized their sharp tails either digging into the endothelium or dragging along and cutting it open. And so in those cases as well, if those cells transform senescent, clear them out, have them be replaced by stem cells instead. And then hopefully we'll be able to positively impact those patients as well, the Lp(a) patients. The outcomes have to be shown, though. So I get a little bit ahead of myself because I can visualize it. And then
That's
I think
okay.
But I hope that we'll be able to impact both of those populations at least, and then have this be a thing of the past.
It's amazing because, the whole CRISPR approach was inspired by rare diseases, right? So who could have thought that rare diseases that usually don't get much funding would be an inspiration for one of the most common disease that kills, a ton of people, but yet they were. But the whole concept of you sort of being able to vaccinate individuals and they would literally melt plaques, not 100%, but pretty significant amount. 40% is nothing to sneeze at, is just mind blowing. So can you shed some light on, the human trials? How far away are you guys from the phase 2, let's say, human trials,
Yeah, if everything goes right and going in the orphan drug designation of familial hypercholesterolemia, so that would be a seamless phase 1/2. And so we would be conversing with the FDA constantly. We would be in direct contact. And so we're hoping to be able to get out of phase 2 by 2029. And that's assuming a lot. That's assuming we have investors who like our primate data. We're testing 3 separate diseases. So atherosclerosis, metabolic disease, and pulmonary hypertension. And so we expect to see positive outcomes in all 3. And if that's the case, then I don't think we'll have a trouble with funding whatsoever. But that is still assuming a lot. So we have to get the investment so that we can go through the preclinical pathway and then initiate that orphan designation. I do have a feeling that the FDA is going to be on board with this based off of the meaningful outcomes across the entire body that we'll be able to show. Hopefully the FDA application is accepted for orphan designation, because that is up to the FDA itself. We have to argue effectively. But yeah, 2029, long story short.
Well, it's not that far away. Now, you mentioned application in metabolic diseases. So how does this approach work in metabolic diseases exactly? Like, can you cure diabetes, or, what exactly, or potentially hope to cure diabetes, right? Type 1, type 2, how would that work?
Yeah, the glucose tolerance and insulin sensitivity tests came back perfect. So it was actually a recovery back to a much more normal functional level. And once you have type 2 diabetes, you can make it go into remission, but it'll never go away. It's very easy. Yeah. And so we can potentially get those biomarkers in a healthy range Not to say that we will cure type 2 diabetes, and I've been informed that I have to be very careful with that.
Right.
So that is functionally based off of the senescent adipose tissue. So it's pro-inflammatory in all cases. And in the gut and the adipose tissue, which is a major signaling organ, thankfully we're starting to recognize it as its own organ. If you have those pro-inflammatory senescent cells gumming up the works, there's going to be a lot of dysfunction. There's going to be a lot of signaling, aberrant signaling, incorrect signaling. And so by clearing out those senescent adipose tissue as well, which is majorly vascularized, there's going to be this reversal of glucose intolerance and insulin insensitivity back to a normal level. That's in mice once again. So we'll be measuring that in diabetic primates, and there is major reason. Our collaborator is so excited. There's major reason to believe that we will see the exact same thing based off of the mechanism itself of senescent endothelial cells.
Interesting. So because, we in cardiology now in the era of weight loss medications that once again proven to be paradigm shifters, right? Taking cardiology and cardiometabolic approach beyond weight loss, mostly because we finally realized there is something called EAT, which is ectopic adipose tissue, which is this very, zombified, if you will, type of tissue that could actually invade cardiac muscle itself, cardiac myocytes. And one of the rapidly rising forms of heart failure is so-called, what I call fat heart syndrome, right? It's heart failure with preserved ejection fraction. So, very typically, people call it obesity-related heart failure or cardiometabolic heart failure. That's, this is what we see. It's a huge epidemic. It's of ginormous proportions, super difficult to treat because the standard medications don't really work. But then came SGLT2 inhibitors, which sort of shifted heart energetics a little bit more toward that ketogenic type of, energy production. And then came, of course, the semaglutide and tirzepatide. And that really showed people that there is more than just weight loss, because some of these outcomes were not really weight loss related, so there is something else. So do you foresee, do you have any trials, any in the pipeline, any pilots that are specifically aiming for this category of heart failure, the one with, sort of obesity or preserved EF, as it's called?
So that's a really good point. And that is our strategy, actually, is to get initial approval with FH, and then you're allowed 7 years of operational exclusivity on the back end. That means nobody can touch your market. That way, we can expand the indications that we're going after into potentially heart failure with preserved ejection fraction. We're hoping to prevent ventricular fibrillation at the same time. A potential collaborator, Northwestern, he's going to be studying this eventually in mice models while we're doing the rest of our work. But we're expanding out, absolutely. Metabolic disease, as many of these diseases that we can treat as possible, because if people have to go off label to get this, the price is going to be higher. We don't want that. We want to have this approved as broadly as possible so we can help the most amount of patients that we can. It would be a darn shame if this was only relegated to FH patients. And so I foresee us being able to expand this far and wide. And actually the downstream too, we can produce this eventually in a room temperature stable format without the need for refrigeration. So we can get this into deserts, into jungles, into third world countries where refrigeration is the inhibitory or the rate limiting step. We're trying to get this into the hands of everybody. And that's part of the strategy of virus-like particles. They're a phenomenal little vaccine, and they're safe, darn it.
It sounds phenomenal. And I keep on thinking, that I'm beginning to feel really sad for Brian Johnson, who is doing so much to keep himself not to die, and is so motivated to do this. And yet, maybe in 5 years or so, maybe in 6, you'll come out with that vaccine and all of those. I mean, certainly all of those other things are very important because, you could, unfortunately, through stress exposure, obviously environmental exposures, poor diet, and terrible lifestyle, the endothelium will continue to be leaky regardless of the vaccine. You can override it, but at least it will give people an opportunity to start early and to go into that preventative route and, truly longevity vertical. I have to say that I am very, very intrigued by this. So when do you expect your primate study to be published? How soon to completion of this? Like those three buckets, right? So atherosclerosis, metabolic disease, and pulmonary function. So will you be sort of like when that project will result?
Yeah, we're fully funded for 6 months, and that's going to be measuring. Well, we expect to see a downward trend within the first 3, and then overt plaque clearance
Mm-hmm.
within the study duration of 6. Same thing with the metabolic disorder recovery. Those biomarkers, we expect to see effects within 3 months. Pulmonary hypertension, that's a little bit more unknown to me.
Yeah, it's harder.
We'll be measuring multiple different outcomes though as well. But we have the rare opportunity to extend this into the first primate lifespan study, which might not be as meaningful sounding, but the FDA does not recognize yet aging as a disease. And I understand from a geriatric care perspective, aging is not a disease. This is more of an FDA standpoint. We need it to be a
Mm-hmm.
disease so that we can target it specifically. If we can have a clinical trial against aging, that would be incredibly powerful. What lacks at this stage are data. If we can have a primate lifespan study, a human relevant lifespan study in a primate model showing lifespan extension in a meaningful way after a senolytic intervention, that could change the FDA's mind. And I forgot to mention these are already geriatric diabetic primates, which is why this is possible. We could do
Mm-hmm.
this in the next 5 years as opposed to having it take 20 to 30 years that it normally would. So we could extend this after the 6 months. We have the option. We just have to gain sufficient funding to achieve that. And
To achieve that.
yeah, the impact could be enormous though. So I'm very hopeful that we'll be able to secure those morally aligned, mission aligned kind of investment partners along the way.
Wow. So the primates, usually these geriatric diabetic primates, what's their life expectancy on average? And, how much of an additional life expectancy you think they may derive, if the vaccine intervention is successful?
Yeah, in the control group, we don't expect many of them to last more than 5 years longer. In the
Mm-hmm.
vaccine group, even in the diabetic case, which is unfortunately a major reduction of lifespan, we do expect to see years of life extension, even in the disease case of diabetes, which outside in a healthy case, we don't know exactly what that would extend to. 5 years, we just don't know, and we'll have to
Mm-hmm.
find that out. But it would mean a human lifespan, and that's probably not something that we'll be testing in anytime soon.
Exactly. Exactly. But to be inclusive, there are other measures, of course, that help people to clear out senescent cells. They're not as robust, perhaps, or as effective as vaccination against a specific marker that those cells are on. So, as a biologist who's been in this area for nearly 15 years, what are your top three that you typically look at as an opportunity to lower the burden of senescent cells, whether it's endothelial or, let's just say, across the board?
Yeah, I always shy away. This is going to sound crazy, but I don't even like taking Advil. I have a feeling that there's a lot of good that senolytics can do. I know that there are a lot of natural products like capers, for example. Capers, if you have them and they're not
Capers
as
are amazing.
right. And as long as there's not too much salt content there, because then you're doing damage by the salt. But many, many natural food products that you can eat that are not as potent with this senolytic strategy, because the worry is there's going to be some damage somewhere, and it's just to this low degree that we can't detect initially. Kind of like how smoking
Hmm.
will kill you over 50 years. It won't kill you immediately. The same kind of thing could be happening with a lot of these different longevity nutraceuticals, but we just don't know. I think for some people it'll be incredibly beneficial, maybe the Brian
Hmm.
Johnsons or the Andrew Hubermans, but the blood tests are required to confirm it. And so if I were going to take anything, I would first get a baseline measure of a blood panel, complete blood panel, all the biomarkers that I could, and then just start testing. If I see a downward trend in my liver function, okay, let's knock that off immediately. Let's go back.
Right.
But I would start with
Yeah.
natural foods first before I started opting for supplementation.
Yeah, for sure. And, we always tell our patients their foundational diets, like anti-inflammatory Mediterranean style diet, high phytonutrient content, which of course, improves your nutrient sensing, clears out your senescent cells. There are interventional diets, and we use something called fasting mimicking diet, which, they are patented. They have shown in animal models and even in humans that autophagy is happening, that senescent cells are getting degraded. So, they do have specific protocols for that. So this is, it's a mildly ketogenic approach, but it goes a little bit beyond that. So, I think there is a renewed interest in various modes of, fasting and caloric restriction, and how those factor into hallmarks of aging, because it's such a powerful, multi-hallmark switch, if you will. But then comes a vaccine like yours, and it could really augment what people are doing, and potentially reverse, some of these dreaded, non-communicable chronic diseases that we're dealing with. I mean, amazing. David, you're a person to watch. I will, keep my eyes peeled on, publications. I'm sure there will be some preprints that announce these results, and it'll be fascinating for sure to see the effectiveness for plaque reversal, plaque regression, metabolic, augmentation, and certainly heart failure data seems to be very, very compelling, even though it's in the animal model. So the future is bright for longevity. I love it. Any parting words for our listeners?
Let's see. Keep going. Keep going. Sometimes the world seems its darkest, but just keep pushing through. We've got this. We will get there, and we'll get there together, I think. And thank you again, Dr. Druz, for having me on. This has been a lot of fun.
Yeah, the same here. I learned a ton, and I can't wait. Make the vaccine happen sooner. Take care. Thank you for tuning in to Own Your Heart Health with Dr. Regina Druz. This podcast is powered by Holistic Heart Centers. If you enjoy the show, please rate and review us on your favorite podcast platform. To learn more about our services, visit holisticheartcenters.com and subscribe to our YouTube channel. The link is in the show notes. See you next week.